is done. So today we very briefly discuss about several different methods that are used to increase the essentially time scale that can be reached as well as the size scale that can be reached by classical MD simulations. So what I'm describing here is on biological systems, but that's true for any molecular systems. Usually you are interested in system sizes and time scales that are much beyond what is reachable nowadays for classical MD simulations. For the case of biological systems, we would like to include the natural environment, usually membrane, solvent, ions. Many systems work as assemblies. You have to include several components, several different proteins working together. You are interested in simulating the whole organelle or the whole cell in order to be able to understand how biology works. But actually, that's the size problem. That's comparatively or relatively speaking the easiest problem, because as you go to larger and larger computers, you can have much larger system sizes. That's something that can be distributed very efficiently on multiple processors. So as we get access to larger computer clusters, we can think about running larger and larger simulations. But the time scale problem is much less trivial, because this is a serial problem, so you have to take the first time step, complete that before you can take the second time step. So we only can rely on algorithms that can do this faster, that can do all those interaction calculation, force calculations that we need much faster using reasonable approximations. But toward this, people have been also thinking about using different resolutions, how to describe the system using sort of less number of particles, softer potentials that allow you to take longer time steps, as well as using bias simulations in which you don't have to rely on thermal fluctuation and random kind of motion of this particle to leave the room. So you can actually go in and accelerate certain molecular events by adding forces, by designing a set of forces that actually can push the system in one direction by applying bias potentials. And that's actually another thing that we will talk about today. So this is one example. This is a biological system, ATP synthase. So this is the protein that produces ATP in your body. And it produces, every day you produce your weight of ATP using this machine. It uses proton gradient across the membrane as a sort of source of energy and generates ATP. And the way it works is actually it has multiple domains involved in it. One part is sitting in the membrane, sensing the proton gradient that goes through this and causes a mechanical rotation of this part that is, again, used up here in the catalytic side of this protein to produce ATP. Just as an example of a system that is composed of about 500,000 atoms, this is the minimum size that you need to simulate to get some understanding about this protein. And the time scale of this phenomenon, one cycle takes about 10 milliseconds. So it's a very, very difficult process. You can't really rely on equilibrium simulation at the level of atomistic resolution to get a reasonable answer for this. So again, this is another thing that I already pointed out. So it seems that every 5 years or so, we have had, fortunately, an increase of about an order of magnitude in the number of processors or number of cores, I should say these days, that are available within supercomputing facilities that are available to us. So now we're going to have this guy with hundreds of thousands of processors. So that is very nice for the size scale problem, because then you can break the system spatially into little pieces, send them to thousands of processors, and let them work together. I already also mentioned the sort of specialized hardware developed at David Shaw's research company. They called it Anton because Anton was the name of the person who discovered the microscope. So they call this machine also Anton because they consider it a computational microscope with which you can look how molecules move, essentially. So they call it Anton, and actually a copy of this machine is available at Pittsburgh Supercomputing Center because many people wanted to use the same hardware and run very long, microsecond-long simulations. So the company itself has several of these, actually next generation of these machines, and they have been able, as I pointed out yesterday, to run even millisecond simulations at atomistic resolution. But we also have access to this machine for longer simulation. But again, otherwise, most of the development in this area have been addressing the size scale problem, but the time scale problem remains to be the most challenging issue. Again, I mean, if you have fast hardware, specialized hardware that can do these calculations faster, you can expect to have a longer time scale, but by just increasing the number of processors, we cannot handle this problem. So one of the pretty common method that has been actually used, and is extremely useful if you use it carefully, and you know what you are doing in molecular simulation, is the so-called bias simulation. In one version of it is known as S-steered MD, or SMD in short. We have actually a little example of this kind of simulation in your tutorial that you might want to give it a try. Now that you know how to run an AMD job, it would be pretty trivial to set up an SMD simulation and run it. And the idea is coming for example, from single molecule experiments like atomic force microscopy, in which you apply force to a molecule to see how it behaves under the effect of this force. We do exactly the same thing. So as we discussed yesterday, we have the force field that generates based on natural interaction of the atoms and the rules that we have defined for general interaction between these atoms. You can calculate those forces, but in addition to that, we can add forces to the system. We can have a sort of constant kind of force applied to one atom or a group of atoms, or we can have a sort of variable scheme in which we change the amount of the force that we are applying to the system, increasing the forces slowly until the system starts to do what we want it to do. Again, going back to the example of me, you are interested in how I leave the room from here. So if you want to wait for me for my random fluctuation, it might take years for me to decide to go that way. But if you start pulling me by an additional force, external force, gently toward the room, so I mean, as soon as I maybe randomly reach this region, this low energy path, then I start moving toward the exit door. So you bias my motion, my random fluctuation, toward certain target that you have in mind. So you can do this to induce different kinds of phenomena in your molecular system. So you can, for example, in case of proteins, we can induce binding of ligands or unbinding of ligands from their binding site to see how they unbind, what is the pathway, what residues are involved in causing the barrier against this. We can do unfolding experiments. This corresponds actually to what has been done experimentally. You can pull the two ends of your protein, try to unfold it and see how it unfolds. You can apply surface tension. For example, if you have a membrane system, you can actually bias it and pull on it and cause a tension and see how the protein that is inserted into the membrane reacts to this tension. If you design your forces nicely, so you have an object that turns during its function, you can have tangential forces on several groups of atoms, different directions, different magnitude, to induce torque and rotate part of the molecular system. We have been using this method to induce pressure. For example, you have a membrane in the middle. Water molecules on the top are pushed against the membrane. Water molecules at the bottom compartment are pulled away from the membrane. That way you can generate hydrostatic pressure very easily in your simulations. I mean, these are things that you might be also interested in doing if you're doing material science type of application. We can induce large domain motions and any conformational changes. So essentially things that would take a long time to happen in equilibrium simulations, you can do much faster. Going back to the problem of time scale. So of course, then you go into the non-equilibrium type of calculation, a non-equilibrium regime, and there might be a lot of hysteresis involved in this, and that's something that you should be aware of. So the way we apply the force in NAMD, which is your example, we have two protocols. If you know how much force you need to apply to see the phenomenon in your limited time scale, then you just apply that force. But usually that's not the case. You don't know whether you have to apply 5 piconewton force on me to see me leave the room in 20 nanoseconds, or you need to do 500 piconewton. So that's why we use a force variable scheme in which we increase the force. It's like you connect the spring to me, and you pull the other end of the spring at constant velocity. As you increase the length of this spring, I feel more and more force on me until I start to cooperate and follow the direction of pulling that you are doing. So this way you don't know, you don't need to know how much force you need. You keep increasing the force until something happens to the system. And as I move and cooperate with you, because the spring is now become shorter, you decrease the force also automatically, and you decrease the force and you don't apply unnecessarily large forces on me, because I'm now, this part of the path is pretty low energy, pretty flat. You don't need to apply any forces. So by monitoring the force profile that you are applying during your simulation, you also get an idea about the position of the barriers. Where you need to apply large forces, there must be something preventing me from going, either entropically or there is a potential energy barrier there. And where I cooperate, you see that the force drops. So you can see what are the major barrier positions of the barriers and what molecular features are responsible for those. So far everything looks great. The big problem is that if you pull too fast, which is usually the case, then instead of taking the correct pathway that you're interested in, this is the pathway you want to be able to describe. If you pull on me too fast, then I start going over the table. I can even cross this barrier, this unnatural barrier, which is along a completely wrong pathway. So here you will see usually an overestimation of the barriers, because you are not waiting for me to explore the configuration space and to find the best pathway to do the transition. That's usually the case. If you do pull it way too fast, then the pathway will be completely unrealistic. But if you do it carefully and you keep in mind that there is always the danger of seeing unnatural and molecular behavior, then you can really use the information you get here to explore the pathway. For example, you might be pulling me in that direction. Maybe in the beginning I go over this table here, so the initial barrier that you see is not natural, but then after this point I might follow the correct pathway. So at least actually a large part of the pathway that you describe by this method will be real. And then you can go back and actually re-examine these points that you have explored using the method of SMD by equilibrium simulations. You can take these snapshots here and try to simulate it and relax it further with equilibrium, and maybe you will find a configuration where I relax to a low energy configuration. So it allows you, instead of saying, no, there is no way I can look at this event in the simulation, forget about it, I can't do it. Yeah, you can actually explore it, you can perturb it significantly and get something, but you have to be very careful in terms of how you interpret, because there will be a lot of hysteresis involved in the process. So that's another sort of one important, very useful type of method that you can use. Now, can we get something quantitative out of this? Of course, qualitatively you get an idea about what the pathway might be, where the barriers are, what is causing those barriers. But then quantitatively, can we also get some information about the free energy associated with the process? So that's really the ultimate goal for us. So this is one example in which you are looking at a movement of a molecule, conduction of a molecule, a sugar in this case, through a channel. So if you wait for this process to happen on equilibrium, you need to run several microseconds or probably millisecond simulation. It doesn't happen very fast. What we are going to do is to apply a force to the center of mass of this molecule and then pull it in along the z direction. The z direction is actually going down this way in this particular example. And the force is distributed among different heavy atoms. As I told you, we usually don't include hydrogens. Those are too light. So based on their mass, the force is distributed between the different heavy atoms, and then you can see how much force is necessary to pull this molecule through the channel. So now, if you look at the displacement in time, this was a rather fast simulation, 1.5 nanosecond. A few years ago, we did that. That doesn't matter. But what I was telling you about the force profile is shown here. So you can see that here it's reaching one barrier region. There is another major barrier region that prevents the sugar from moving forward. So by just looking at the force profile, I get some idea, qualitative idea about the position of the barriers against the conduction. So, but as I pointed out, I would like to be able to derive the free energy profile associated with the transition of this sugar molecule through this pore. That's my goal. So I know from second law that the work that you're doing to the system, you're applying the force to the system and the sugar is being displaced, you can calculate how much work you're doing to the system. But this work, because you're in a non-equilibrium regime, is going to be, actually in macroscopic world, is always larger than the free energy or equal to the free energy that you want to calculate, right? So that's always the case in macroscopic world. Now let's look at what happens in microscopic world. In a molecular world, things are a little bit different. So this is so we are talking about the so-called Jarzynski's equality, which was based on a few other theories. So you are interested in the transition between two states. This can be described as a time-dependent variable. So you go from initial state to the final state. During this transition, there will be exchange of work and heat. And you are interested in the free energy difference between these two states. That's what you want to calculate. Now, as I pointed out, in macroscopic world, when you are in a non-equilibrium or when you are doing work to the system, the work is always larger than the energy. When you come to the microscopic world and you repeat this experiment, you just pull this sugar from this position to that position, and you repeat this several times, and see how much work you have to use every time you complete this process. And then you plot the histogram of the work that you're doing to the system. If you look at the probability of the work, and you look at the average of the work, this is going to be the average of the work is going to be larger than the free energy that you are interested in. Because some of the work goes into hysteresis. You're pulling too fast. I'm going over unnecessary barriers. So you will always use too much work on the molecular system. But actually, there are certain rare events or cases in which the work that you are doing to the system is actually less than the free energy difference. And that's because there are cases in which I'm actually already going out because of all the thermal fluctuation of the system. It's doing the process for you by biasing my position along this pathway. You're just slowing me down. So the work that you are doing to the system will be less than the actual free energy difference of the process. And that becomes actually really interesting. This sort of rare events, most of the time, you're still spending too much work, but there are cases in which the work is going to be less than the free energy that you want to calculate. So for those cases, then, instead of comparing the average of work, if you look at the exponential of work, then you can see that the exponential of work would match the exponential of the free energy difference that you have. Because this exponent is dominated by low work values. It's a negative exponent here. So these guys in which you have minimum work used to induce the process will dominate the average of this exponential. So then it has been shown, several nice theories, that despite the fact that the average of the work is larger than free energy difference, the average of the exponential of the work will match exactly the free energy difference that you want to calculate. So if you repeat this simulation several times in order to be able to sample these rare events, then in principle there is a formalism allowing you to calculate equilibrium properties from this non-equilibrium type of calculation. This actually has been proven experimentally. They pulled polymer of nucleic acids using fast non-equilibrium type of AFM or optical tweezers. I don't remember exactly the experiment. So they measured how much work is used in non-equilibrium cases, and they did it also very slowly, sort of quasi-equilibrium type of measurement, and they showed that these two are within 0.5 kilocalorie per mole for such a complicated process. So it has been experimentally also proven. But again, the point is that you have to repeat this. In those experiments, they repeated this several hundred times. And this is unfortunately not something that we can do in our simulation. So we still have a problem with sampling. Either you have to have very, very slow simulation, or you have to have many, many, many fast simulations in which you induce the process in order to be able to sample this part of the distribution. So the sampling problem remains there. It just has been sort of reformulated. Again, we need to have repeated non-equilibrium experiments or simulations in order to be able to derive equilibrium properties. So that's a very brief introduction of the method of SMD. Again, there is one example. If you find it useful for your research or you're interested, use that to see how to set up forces. So now let me actually go back to the main subject of the day, which is now another way of dealing with the problem of time scale, or to some degree size scale, is to coarse-grain the molecular system. So the method of coarse-graining has been, for biological system, has been pretty successful for membranes and lipid structures, lipid bilayers, vesicles, micelles, and that's why I'm using membranes as a sort of example for my talk. Let's look at some of the properties of membranes. So membranes are formed by two layers of lipid molecules. These lipid molecules have a head group that is charged or very polar. It faces the water. And they have the tails that are not hydrophilic. They don't like water, so they look away from water, and they find each other in the middle. And that's how the bilayer forms. So you essentially have two leaflets formed of lipid molecules. If you look at the diffusion of lipid molecules, for example, within the same monolayer or same leaflet, that would be the diffusion constant you have, 10 to the minus 8 centimeters squared per second. Just to give you an idea, it's about 3 orders of magnitude smaller than water diffusion. So that translates, this is the diffusion of water. So that translates in a very slow lateral diffusion of lipid molecules within the membrane. It doesn't mix. So on the average, if you will have about 6 angstrom in 1 nanosecond, and you have to have hundreds of nanoseconds of simulation in order to be able to see when lipid molecules exchange their position. This is one example of the slow processes that we are interested in simulating. So now, this is an example of an atomistic simulation of a lipid bilayer. So we have all the atoms represented explicitly here. And you can see this is a 5 nanosecond simulation, actually. And you can easily see that lipid molecules The phosphorus atom of the head group is shown using large spheres. You can really see the position of each lipid molecule here. They go up and down a lot, but they don't go anywhere as far as lateral diffusion is concerned. So that has caused a lot of problems because, for example, we cannot see mixing of lipids. We don't know if you add cholesterol to this lipid bilayer, what happens to it, how fast it moves, et cetera, et cetera. It has been actually difficult. So that's why we, or actually this is not a new thing in lipid bilayers. For many other systems, people have been doing this. But in biological systems, people started to think about coarse graining, the description of this molecule. So instead of showing all the atoms or using all the atoms in this description, we are going to group these atoms based on a sort of chemistry, based on their properties, how they interact with their environment, and use a reduced representation for this molecule. So in a way, you can even consider this atomistic representation already a coarse-grained kind of representation if you compare it to a quantum mechanical description. In a quantum mechanical description, you have the nucleus and all the electrons. When you come to atomistic description, classically, then you will have one particle for the sponge, the nucleus, and all the electrons associated with it would be one particle. Now we do it another level. We are going to group together a few atoms and represent them as one bead, one particle. So that's really the essence here. So you have to have some idea how to do this mapping. For a standard residues, lipids, proteins, nucleic acid, it has been done by others. People have tried a lot of different ways, and we have a sort of rather standard or highly tested protocol to do this mapping. For example, for the case of lipids, we usually have a positive charge here, which is this blue guy, a nitrogen with a few groups connected to it. This would be the phosphate group, which is usually negative charge. Then we have a glycerol moiety, which is not charged, but is highly polar. That is represented by this group. And then the tails are usually hydrophobic hydrocarbons that are represented by 2, 3, or 4 beads, depending on the length of the tail. So that sort of makes sense in terms of the properties of these beads. These beads have different properties. Some of them carry charge, obviously. Some of them are polar. Some of them are completely non-polar. Some of them can form hydrogen bonds. They have capability of forming hydrogen bonds, accepting hydrogen bonds, or donating hydrogen bonds. So they have tried to include some of the chemical properties that are important for the structure and dynamics of these molecules into these beads. So there are a lot of coarse-grained models out there, but the one that we use, just as an example to give you an idea how they are done. So the one that we use is based on Martini. That's the name of a force field, coarse-grained force field that was developed initially for lipids and then later for proteins. And there they have a certain number of beads used to represent lipids, proteins, and water. Usually, and again, we talk about this in more detail later, so usually what they do is to map 4 heavy atoms into one bead, just to have roughly the same kind of mass for all these beads. You don't want to have sort of large variation in the size of these beads, because then the dynamics, there will be a larger disparity between these particles. So that looks nice. So can you tell me what you gain by doing this description? Do you want to guess? Obviously, we are reducing the number of particles in the system. So that reduces the number of degrees of freedom in the system. That already helps. So what else do we gain by this description? How does this make our simulation faster, in addition to reducing the number of degrees of freedom?
Less.
Less?
computing the interactions would be much less.
Absolutely. So that's this. But actually, do you remember why are we taking 1 femtosecond time steps in MD? Yeah, because we have those light atoms' fastest degrees of freedom. So, exactly. So now by going to this huge, bulky thing, so you have much softer potential. So your vibrational frequency is much smaller, meaning that it doesn't matter. I don't have to take 1 femtosecond time step. I can take 2, 10. Actually, we can take 20 or 50 femtosecond time steps for this description. If your description is different, if you take, let's say, I take 1 bit for the whole head group here and 1 bit for each of these tails, you can probably take even larger time steps. So suddenly, you gain a factor of 20 or 50 by just changing the potential, because these are governed by much softer potentials. Plus the sheer number of these interactions have been reduced, and then you gain between 1 to 2 orders of magnitude speed up in your system. Very important. This is extremely useful. You see a lot happening in your simulation, but very important, you are losing the chemical details, obviously. If it's important to have this particular arrangement of these oxygen atoms here for anything, for interacting with your protein, you will not have that description here. So of course, you will lose chemical details. And one thing that we do here is to see how we can go back and forth. I will use an example to show you how we can go back and forth between coarse graining and atomistic representations to take advantage of both aspects. Okay, so I guess, yes.
How many sites do you have for the charge in these coarse-grained particles?
For each particle, we have 1 site. These are still a sort of 1 site. So we will see for the case of water. I think actually maybe that's a place I can talk about this. So you can imagine one important property is the dipole moment of some of these molecules. So best example would be water. So I mentioned that in this mapping, you will map 4 heavy atoms into 1 coarse-grained bead. So we do the same thing for water. So you can have coarse-grained water in the environment. Actually, you need that in most cases. So 4 water molecules will be packed into 1 bead. So now you had a water that had a nice dipole moment. Every molecule had a dipole moment. It could fluctuate. Now you are going to put 4 water molecules together into 1 bead, which is going to be neutral. So you completely lose the dielectric effect of water in this description. So more advanced revisions of coarse-grained models have started to include a dipole moment on top of this sort of single particle description for these coarse-grained beads, but that is still being tested. And I don't think it has been implemented in any standard package yet that you will use. So what you will do today will be using essentially water that doesn't have any dielectric properties.
So all these particles are spherical, or do they have some shape?
These are spherical. I mean, that's also misleading, the figure I'm using. So these particles mostly are spherical. Thanks for pointing that out. But again, people started to introduce anisotropy in the shape of these particles. It becomes much more complex and more expensive to calculate because you have to keep track of this orientation as well. But that's another thing people started to realize. I mean, it's usually the case. So you start with a very coarse description, and then you realize if you bring back a little bit of the full description, it performs better, of course. And that's something that people are also playing with these days. Yeah. Yeah, so this dipole moment and the lack of dielectric effect is actually a major problem. So for example, dielectric effect of water is extremely important for charge-charge interaction, right? I mean, you screen the charge-charge interaction usually with water, and you completely lack this in coarse-grained models that are commonly used. So if you are doing a coarse-grained simulation in NAMD, then you have to turn on the dielectric constant, and you explicitly specify a dielectric constant of AD. So that way you will screen charge-charge interactions in a very coarse manner, but still that gives you some of the effects that you will have from a real water. Are you with me? So usually when you have explicit water, atomistic water in your simulations, the dielectric effect should come from those explicit water molecules. They fluctuate, they polarize in response to the environment, so they will give you explicitly the dielectric effect. When you do coarse-grained water, you will not have the dielectric property because it doesn't have a dipole moment. Therefore, you have to implicitly include that dielectric effect. So there is a command in NAMD configuration. You turn on dielectric constant, and you specify what dielectric constant you want to use, 80 in this case, or 78. And that affects all Coulomb interactions that are calculated in your force and energies. Everything will be scaled by 80, essentially. So you might argue that in some places, like inside the protein, when you have two charges sitting next to each other, there should be no dielectric effect affecting this charge-charge interaction inside the protein. But when you apply such implicit type of general dielectric constants in your simulation, you will affect everything in a very non-specific manner. But it's much better than not having any effect. Okay, so I think this slide we already covered, but let's talk about this a little bit. So yes, in all atom MD, because of those fast degrees of freedom, we need to restrict ourselves to 1 to 2 femtosecond. That limits the overall time scale of the process that we can simulate to hundreds of nanoseconds or microseconds. When we do coarse graining, we reduce the number of degrees of freedom. Fast frequency motions are removed. Hydrogens don't exist. We have much smoother and softer potential surfaces, and then we can take longer time steps and reach microsecond or even longer time steps. So as I pointed out, usually we use between 20 and 50 femtosecond, depending on the coarse grain model that you use, and that allows us to easily run tens of microseconds for the molecular system. Okay, I guess this is all, again, summarizing what we have talked about already. And again, this method for biological systems has been successfully applied to lipid systems. And the reason is that in lipid molecules, the main distinction that you need to make in order to capture the structural and dynamical properties of lipids is the distinction between hydrophobic tails and polar charged head groups. That's a major distinction between these two. In order to form a bilayer, you just need to have a bunch of guys that are hydrophobic, a bunch of guys that are hydrophilic. The details of the chemistry do not matter much. Whether you have a specific hydrogen bonds oriented that way or this way, it doesn't make much difference. You will get a bilayer structure as soon as you have two kinds of beads. Some of them are hydrophobic, some of them are hydrophilic. So whereas when it comes to the protein structure, you have 20 different kinds of amino acids. Every amino acid has a specific properties: hydrogen bond donor, accepting, being polar, having charge, having different shapes, different sort of stereochemistry. So all these things matter for protein structure. So because of all these specific things that are important for protein structure, this method has not been very successful in describing protein, or I should say as successful in describing protein structures and complicated molecular systems. So if you have a sort of ordered system in which you can see patterns of interactions that are nicely can be nicely distinguished from each other, then you can really nicely take advantage of the method of coarse graining. If not, then the method will not be able to describe important aspects. The same is true when you compare atomistic and quantum kind of calculations, or classical and quantum mechanical calculations. So if you have processes, phenomena that are not too sensitive to electronic restructuring or electronic structure, then you can describe them at the level of classical description. If you have things that are highly polarizable, conjugated systems, excited state kind of calculations or problems, then you have to have a treatment that captures those. So the same is true here. So now this is actually one example. So I showed you what happens if you do an atomistic simulation. Using a bilayer, nothing happens, just lipid molecules just fluctuate up and down. So this is one example of one of the first coarse-grained simulations that was reported by a good friend who is actually one of the leaders in this area, applying coarse-grained models to biological systems. Initially, most of the work was done in lipid systems. You will see how a vesicle forms completely within the simulation. But they also extended the model into proteins, as I put it. He is the developer or the main developer of the Martini force field. He says one of the reasons he called it Martini is that he likes Martini, but he also has an abbreviation for that. So initially you have a bunch of lipid molecules. These are lipids that usually form liposomes or sort of spherical bilayer-like structures. You have to be a little bit careful about this time scale here, and that's something I will talk about a little bit more later. So the lipids that will form the external layer are shown in green, different shades of green, dark and light green. And lipids that go into the other layer are shown using orange and red, or the other way around. So you see that initially they're completely mixed. You have water, coarse-grained water also here, but it's not shown. Oops. So let's watch this and see. Please. So this is not really a real time scale that you can see here. I will talk about this as actually one problem associated with coarse-grained simulation. So you can see actually that nicely, it sort of, you see, they are sort of going into different partitions and compartments here. Some of them come to the surface. Some of them are aggregated in the middle, forming the inner layer. You will form a sphere. Inside you have water. Outside you have water. They are not shown, so you can see. And then at the end, it's going to cut through this, so you can see the structure. So they form a very nice spherical bilayer structure, which is known as a vesicle or a liposome. So now we cut through inside. There is water here, and so forth. So this kind of process can be done very nicely with coarse-grained simulation. So there has been no atomistic simulation that can even describe partially this process so far. Okay, so I think I might. That's much better. Okay, so this is what we discussed so far. So for the case of lipids, you can actually do the mapping depending on the architecture of the system. For the case of lipids, there has been some standard ways of doing that. We have this in NAMD, and you can use the program that is the interface that we have in VMD to convert your molecular system from atomistic description to a coarse-grained description. As I pointed out, there are different kinds of coarse-grained beads that are used. There are several different groups that are actually working on this. The one that we will use is based on the work of Marink and his colleagues. And as you can see, they can have positively charged, negatively charged, and polar and non-polar beads. Water is described usually as a polar bead, which is not charged, doesn't have a dipole moment, and is composed of four water molecules. You can do the same thing for proteins. So proteins are composed of residues that form a polymer. So you have the backbone, which is the same for all residues almost. And then for each kind of residue, you have a different side chain. So you can describe every residue in a protein. For example, this guy here, a tyrosine. You can have one coarse-grained bead describing the backbone, and then you can have one coarse-grained bead describing the side chain. That's the simplest kind of description. The side chains are smaller in some cases and larger. So this is your protein backbone, and you can see the side chains sort of branching out from the backbone. This has been preserved here. So you saw these are the backbone coarse-grained beads, and then you have description of the side chain. Sometimes this is a nonpolar side chain, big, nonpolar side chain, small. You have a fairly large semi-polar side chain here, which is shown using a different color, and so on and so forth. So the same principle applies here, but as I pointed out, things are getting a little bit more complicated. For example, one important thing for protein coarse graining is that the orientation of the hydrogen bonds between the backbone atoms, between carbonate and amino group in the backbone, the orientation of those hydrogen bonds is really important for formation of helices and beta sheets. And because you lose this orientational description in a coarse-grained representation, if you don't apply additional constraint, your protein is going to completely melt. You will lose these secondary structures in coarse-grained descriptions because the orientation of the hydrogen bond is important. So that's one major thing to remember if you want to use coarse-grained models for proteins or other polymers or other molecular systems in which the directionality of hydrogen bond matters. So right now, what we are doing is to either constrain the protein to preserve its secondary structure. We keep the helical conformation by constraining the dihedral angle along the backbone. Or we can actually use some sort of network models in which we the overall shape of the protein will be preserved. So in the network model, what we do is to look at the whole protein. Some of these atoms are connected to each other through chemical bonds, but we define additional bonds connecting every pair of backbone atoms that are within a certain distance. For example, you say if two atoms are within 10 angstrom, I'm going to define the bond between them, an additional bond, and I'm going to use a sort of soft spring constant for that bond that keeps those bonds within the original distance. So when you have a lot of these bonds defined within the protein object, then you can see actually a network of connectivity within that shape that preserves the overall configuration of that object. So that's another way of dealing with this problem in coarse-grained complex. This is actually the better description because you do coarse graining in order to observe large-scale conformational changes. You want to see what happens to your protein when you do coarse graining. So now, if you have a helix that changes its conformation during the process, but you constrain it to remain helical during the simulation, then you won't see that. Whereas using this network description, things are a little bit softer, sort of less specifically constraining the protein structure. You have a better chance to describe some of those large-scale protein conformational changes. Again, as I pointed out, you will use NAMD as a molecular dynamics engine to do coarse-grained systems, but as we will see, the topology and parameterization is largely based on the work that is done in Martini. So the recent version of Martini, this was the original kind of coarse-grained representation. Each side chain, regardless of its size, was described by one coarse-grained bead. Sometimes smaller, sometimes larger. But later, they again going back to the idea of providing a less incomplete or more complete coarse-grained representation. They started having different type of coarse-graining for different side chains. Sometimes you have one bead for the side chain, sometimes you have two, sometimes you have three. For example, this bead, big representation is used for rings here. And then you have something like that for residues that have two rings fused to each other like two. So they have a sort of refined and improved representation for latest revision of the. So there are some references here, so I can make these slides available if you want to dig further into the details. But again, this is just to give you an idea. There are many different ways of doing coarse graining, different potentials, different kinds of beads, and lots of people are trying them and testing them to see which one is performing better. So let's actually use the example of alimenticin to walk you through a sort of example of a coarse-grained kind of simulation. So this protein, this is something that we have done in the lab. This particular peptide is an antibiotic. It forms chemical bundles inside the membrane. It is formed of 15 to 20 residues here. Most of them are hydrophobic. As you can see, they're shown in gray. We have a couple of hydrophilic residues here. This is a single monomer here. When it's added to the membrane, it forms channels in the membrane. And by forming pores in the membrane, it essentially kills the cell because you kill the proton gradient and ion gradient across the membrane. The question is what happens to this and how it oligomerizes inside the membrane. So that was something we wanted to describe. Okay. So yes, that's actually the model in the literature. So people suggest maybe it forms form of 5 monomers, 6, 7, 8. There are lots of data out there, and there are also models about how they orient themselves and how they aggregate inside the membrane. So we wanted to test this with the coarse-grained description. So this is the initial configuration here that we started with. This is an atomistic kind of description that we started with. We placed in a sort of lattice-like structure, 49 peptides, 7 by 7, and 288 lipids. So we relaxed it a little bit. Then we converted everything to coarse-grained models. That's exactly what we're going to do today using a different protein. So you have the tool to convert this to a coarse-grained system. The mapping is shown here based on what we discussed so far. Lipids are mapped like this. A couple of residues for the head group or beads, and some for the tails. And for the proteins, we are using the sort of old model in which we have one backbone and one side chain representation of beads for each residue. So we take it to the coarse-grained representation, then we run it for a microsecond because that's what we can afford using that representation. And then because you see the general aggregation properties of the system at the level of coarse-grained. But at that level, you never see formation of tiny pores through which water ions can go through, because this is too coarse. You don't see water going through, you don't see where the defects in the membrane might go. And in order to characterize those, this is it's not blue anymore. I don't know if it's the battery is out or.
Don't worry about it. Leave the other one.
So once we have something every now and then from our coarse-grained description and simulation, we take some snapshots, reverse coarse-graining, bring it back to atomistic description, and again, that's in principle looks easy. But in practice, it's not trivial. So that's another part of the tutorial that we do here. You reverse coarse-grain the system, make it atomistic, and then examine those snapshots using atomistic simulations. So it's not a real multi-scale type of simulation in which both descriptions are done at the same time, but by going back and forth between coarse-graining and atomistic simulations, you can actually gain a lot of insight about the system. Again, comparing it to what can be done between classical and quantum mechanical description, it's like you want to study an enzyme, you run this atomistic simulation of the enzyme for several nanoseconds, and you take snapshots from these atomistic simulations, and you examine those snapshots using quantum mechanical descriptions and see what happens in the active site, for example. ATP gets hydrolyzed in the active site. I just run my ATPase protein for 20 nanoseconds. Every 1 nanosecond I take a snapshot and just look at the active site and calculate quantum mechanically what is happening in the active site to see what might be a good moment for proton transfer because I have the correct direction of the water molecule and the substrate. So this is exactly the kind of approach that we are taking. Yeah, so we have been examining these snapshots using 20 nanosecond atomistic. So that's again to give you an idea. So when you do a coarse graining, you go from hundreds of about 100,000 atoms to about 10,000 atoms. But again, the main reason of gaining speed is that we are dealing with software potential. So this is again something that I already talked about. This is the mapping, but actually now these are the kind of beads that were used in that particular study. We have charged beads, we have non-polar beads, polar beads, and we have apolar beads. So the names are confusing. You might wonder what's the difference between apolar and non-polar. They are different in terms of the capability of forming connection bonds. Water molecules, 4 of them are bagged into one coarse-grained bead. And for the ions, we do the same thing. So for the ions, the mapping is a little bit different. We put together the ion and the first hydration shell for that particular ion into a coarse-grained. Because if you don't do this, these water molecules will not be able to solvate this ion. These water molecules don't have any dipole moment on them. They cannot solvate the ion. So that's why we include the first hydration shell of the ion in the course. Now about the time scale, actually, that's something that is important, so we should talk about this a little. So as I pointed out yesterday, when you are doing quantum mechanical calculation or even classical simulations, so your potential is based on physics, based on something you can measure. You know from vibrational spectroscopy the vibration of a CH bond is on the order of 10 femtosecond. You design that CH bond using a spring constant that gives you that. You know the mass of this particular atom. When you come to this description here, the coarse-grained description, there is no physics that you can collect. There is nothing that measures what is the vibration between 2 coarse-grained beads, between 2, these things don't exist as single entities. So it's very difficult to gauge the time scale associated with the dynamics of these beads. So one way of doing that would be, let's say, okay, so I can use one element in the system and try to find out what is the time. I mean, in the simulation engine, we can change in the configuration. Instead of using 1 femtosecond, we can put 20 in the configuration file, and that would take longer time step for integration. That's no problem. But whether or not that's really 20 femtosecond or is it representing, I don't know, 100 picosecond, we really don't know. So we have to have a way of calibrating the time scale associated with the motion of these particles and what is happening. So that's a problem. So that was a problem on the movie I showed you. There was a counter showing the time scale of that phenomenon, and if you remember, it was on the order of a few nanoseconds, which is absolutely wrong. So if you do an atomistic simulation on the same system, several hundreds of nanoseconds, you wouldn't see that phenomenon. So the counter was there, what was not really representing the actual time scale of the process. So one way of doing that would be to say, okay, let's look at the diffusion of water. I know how fast water moves, right? So I know the diffusion constant of water. I can run the simulation of a coarse-grained water model and see what I get. And based on that, I know that what is the calibration, what is the scaling that I get. So based on that, I can calibrate the time scale based on water. But then there is no guarantee that this is also true for the scaling of different kinds of particles in the system. Because definitely there will be a disparity between water coarse-grained water and the residual coarse-grained feeds and the lipids. So that continues to be really a very difficult problem in terms of what really the time scale that you are simulating in a coarse-grained representation is. Because there is no easy way to connect that to a real physical system in which you can do measurements. There is no property that you can connect. There is no vibration of spectroscopy for this coarse-grained representation. So the problem time scale disparity is something that you need to keep in mind. Again, if you are dealing with bulk solvents, that might be not a difficult case. You can actually calibrate it against that particular kind of material. But when you have a mix of different kinds of materials, then it becomes very difficult. So the problem is that, again, this might also affect the dynamics of the system. So in a real system, everything is moving in a coherent manner based on well-defined time scales. And you go to coarse-graining description, you might actually make your protein slow compared to water. So there will be disparity in dynamics too. That might affect the overall behavior of the molecular system. So these are
individual components are behaving as expected, like in bulk. For example, so the water diffusion is something that you can, of course, scale.
You can calibrate, exactly.
And also lipid alone.
Lipid, I think if you know the diffusion of lipid molecules in a membrane, yes, you can also have some way to calibrate that part.
Right.
But so then again, for your protein,
so the mixture is a problem.
The mixture is
so individual components are behaving as expected.
Well, I mean, for components, we have ways of calibrating and knowing what is the real time scale associated with that particular.
You can also, I think, model your parameters based on those, diffusion bulk properties.
Exactly, exactly. And that's actually, of course, that's how you can calibrate. You can, if you think it's fast. The time set that you use for the integration is not a real time set anymore. That's really the thing that I would like everybody to take. So it's not real anymore. So what it is, now we have, for some systems, we have a way to find out what it means really, but for most cases, especially if you are dealing with a mixed system, we don't know. So you might have actually, you are now putting together, so just for example, fast water and a slow protein. Now how this would affect the overall dynamic of the whole system is another. So usually these are not really serious issues, but that's something to keep in mind, especially if you are looking at bulk properties for your material, for your surface, or things like that. So for the case of proteins and biological systems, that's a rather minor issue because they are mostly interested in qualitatively describing what large structural changes might take place in these systems. So, and again, another problem that I pointed out, and we should definitely keep in mind very seriously, is the fact that you lose the dielectric effect of water when you go from this description in which you have a clearly defined dipole moment to this single particle neutral representation that has no dipole moment. Okay, so so let's actually look at again. This is just to highlight the different kinds of beads that are used for protein simulation. So this afternoon you will work with the lipoprotein. That's the particle in your blood that carries cholesterol and all sorts of lipids. So it's a protein wrapping around a bunch of lipid molecules, and there is no structure available for that, but using coarse-grained models, people have been able to model the possible structural model for this particle. And that's a very nice example of coarse-graining because it has a protein interacting with lipids, and you can nicely do the mapping back and forth and calculate some properties. So So for those, so we can use actually what is implemented in MD, which is based on Martini. But again, Martini has changed recently, and we have a different kind of presentation for some of the beads. I don't want to confuse you with the details of the kinds of beads that are there, but I think it's important for you to realize that all the interactions, all the specific features that you would like to add to a bead can be done using kind of Lennard-Jones potential type. So if you want something to interact through a hydrogen bond, you can make it hydrogen bond donor and hydrogen bond acceptor interacting in an attractive manner using an additional Lennard-Jones type of potential. That's how it is done mostly coarse-grained models in MD. So we have apolar residues, we have polar residues. The degree of polarity can change. For example, we have P1, P2, P3, P4, P5. So then we have, these are some examples of residues that can actually be donor and acceptor for hydrogen bonds. This is a bead, not a residual atom. So that cannot form any hydrogen bonds. So sometimes actually the size of the system is included. To see, we can have C5, C4, we have subtypes of beads included. Again, depending on what features you want to implement in those beads, you can have really different set of coarse-grained beads in different programs. So yes, I mean, then for the lipid, the same is true. You can have charged, as far as the charge is concerned, and hydrogen bonding capability is different. For the lipids, you can have charged residues that carry charge. You can have polar residues that only accept or donate hydrogen bonds. So all these features are included as additional non-bonded conduct interactions to these beads. So you don't need to know all these details for the particular model that you're using, but what I want you to know is that you can add certain properties very easily to these beads, and by defining additional rules in your coarse bead, if there is a hydrogen bond acceptor and a hydrogen bond donor, they should attract each other through this additional Lennard-Jones potential with these force constant and with these parameters. So that's how it is done in MD. Okay, so now, so this is actually just to give you an idea. So after this, we can have a break. So this is the initial configuration of the system. If you run this very long atomistic simulation, not much happens, but you can see that after about 50 nanoseconds, I don't know what it means in terms of the real time scale, but 50 nanosecond coarse-grained simulations in NAMD, you start seeing aggregation of these peptides in the membrane. Peptide, of course. So all these things happen in your simulation, whereas if you do an atomistic simulation, there's a very little chance to see any of these. Just some examples. So all this is done using the same kind of potential actually in NAMD. We are using bonded interactions, we are using bond angles, integral angles, and non-bonded interactions. So everything is done at the level of topology and parameters, and that's something that we will take a look at after the break to see where these are done in NAMD. And in case you want to change this in NAMD and introduce your own topology and parameters, where you need to change the numbers. But as far as the shape or form of the function or form of the potential energy that you use for this calculation, the integrator and everything is exactly the same. You are just using a different definition for the molecular system. You have less number of particles and different kinds of bonds involved here. The major difference is that you will have additional kinds of interactions, as I pointed out. You don't have an explicit hydrogen bond engineered in these particles. And you just add it as a flag to these particles, and they know where they can form the hydrogen bond to each other, and then they should attract each other more, and so forth. So that's really, in a way, very simple. The way it is done in NAMD, and many other actually NAMD engines that are used for binary cross approaches. Okay, so maybe it was too fast or too much, but again, we have a chance to go back to some of the key points in the second hour and talk about those.